immunology के about the immunology topic. Now we are going to discuss about the immunology and what about the consequences and the significance of the immunology, and every aspect I am going to discuss in this topic. So starting from the immunity, the term immunity means the response to protect ourselves from any pathogen. Whether the response is different types, but main aim of this response is to protect ourselves. So coming on the types of the immunity and how immunity response get arises. So about this, if immunity response arises, the main criteria of arises of these immunity response is first one, that is arising of this immunity due to the mainly the pathogen is entered. And if we are not saying pathogen specifically for the immunity, because any foreign particle, that is antigen, any foreign particle that is entered inside our body, and that is non-self, because it is from the outside environment, right? So it is non-self. So any foreign particle that is non-self for our body, we are going to say that it is immunity is going to be arise for this particle to eliminate this, right? After that, if we want to discuss about the immunity, that is active immunity and the passive immunity, mainly the classification of the immunity, active immunity and passive immunity, and how it is classified. It is classified on the basis of how the immunity get arises, means what is the time of the response, how this response get erased. So in all these circumstances, we are describing immunity into the two parts: active immunity and the passive immunity. Active, that is immunity that is by making the antibodies. Our body is making the antibodies. Antibodies are nothing, they are just like the soldiers of our immune system. They are the soldiers that are going to be fight, that are going to be fought to protect ourselves from the foreign particle, because foreign particles try to invade our body and can be harmful for our body. So prevent this harm to prevent our body from this harmness, the antibody get arises. So this is the active response of our body, right? Now this active response may be of two types, whether it is given by the nature or whether we are acquiring this active response of immunity artificially. So the natural active response is of two types, mainly by the pregnancy and the changing encounter of the antigen. Encounter with the antigen, how antibody is forming actually. And in the active immunity, we are going to take the antibodies. However the antibodies come, it matters by the classification of the natural or artificial. If it is the source of the natural immunity, then the antibodies may become from the antigen exposure or by the pregnancy. Because remember, whenever in the case of the pregnancy, the mother antibodies can, that is mainly IgG antibodies, IgG1, IgG2, IgG3. These are the subclass of the antibodies. However, IgG4 not allowed to permit in the um, plus permit through the placenta, because it have the placenta presentation in the IgG antibody, and due to which it is permeable through the placenta, as well as it have the lowest molecular weight, so that it will be get susceptible and go to the placenta, and from the placenta it pass to the fetus. So this is the two sources of the active immunity of the natural type, that is whether we get encountered with the antigen, means foreign particle, or we get in, and so we get embedded by the pregnancy. During the pregnancy, this antibody from the mother pass to the fetus. Now if the antibodies get inserted inside the body by the mechanism of the artificial method, artificial method, we want to make the antibodies prior to the infection, prior to the pathogen enter the body. How it is possible? Actually, this is the same procedure that we already want to make a army to prevent any so that in any circumstances that are not helpful for our body, means they are going to be harm. So already we have the proper army that is ready to fight with this pathogen. So if this at these circumstances, this artificial vaccination process is used. What is vaccination? Vaccination is the, whether it is the toxic form or the and inactive form or the substituted and attenuated form of the antigen that get exposure to the body, that get exposure to our body. So these attenuated antigen these attenuated pathogen or the killed pathogen, they are not going to be harm our body in such a way. And maybe the different procedure of the vaccination, sometimes, uh, for example, like the cholera is the cause the diarrhea in the body. So the mainly the amount of the pathogens are also matter, whether they are going to be cause the disease or they are going to be cause work as the carrier, or they are not harmful, because the amount means number is also dependent. Suppose if the bacterial count in our body is less than the 10 to power 3 for the Vibrio cholerae, then it is not able to cause the diarrhea, because our for causing the diarrhea, our acidic environment of the stomach is going to be alkaline. But if the amount of the Vibrio cholerae is less than the 10 to power 3, then they are not, then these Vibrio cholerae bacteria are not able to cause the alkaline medium, and hence the diarrhea is not caused by the Vibrio cholerae at that time moment. So this same procedure can be applied to the vaccination process, means you are going to give the exposure less amount of the pathogen that are not able to divide, and as well as not able to cause the disease subsequently, right? And another process that by the artificial medium, by introducing the antigen itself via treatment or exposure to the antibodies itself in the same case. So if you are exposing the artificial acquired immunity, or the artificial active immunity can be achieved by giving that antigen that is harmful for the other pathogen. So it is the same sequence, right? Now if we talk about the passive acquired immunity, so it is further classified into the two types, whether this passive immunity naturally or artificially. Passive means transferred preform antibody. Transferred preform antibody means these passive immunity, in which antibodies are not formed in our body. Antibodies are not formed in our body by this passive process. However, in the active process, antibodies are formed in our body, whether they are due to the exposure of the antigen, outside antigen, or the process of the vaccination. So the antibodies are already formed outside our body, and they are ready antibodies that are transferred in our body. That is the passive process. So mother to fetus, this is the natural method, to placenta, and as well as the cholesterol. Cholesterol is the mother milk, first mother milk. It consists of the IgA antibody, and it have the not in the monomer form. It is in the bivalent, and it have the two subunits, not subunit, one new subunit. It have the two subunit IgA antibody. They are both are joined together by the J chain, right? And artificial passive immunity can be achieved by the immunotherapy. We are using these preform antibodies by the help of genetic engineering. These directly preform antibodies directly supplied to the person that is required to fight with any pathogen. Now, type of the immune response. How the upper immune response is achieved? when means immunity. Immunity is the process, and immune response is the way how they are performing this process. Immune response is the how the way they are performing this process, right? So innate and acquired. Innate immune response and acquired immune response or adaptive immune response. Innate immune response is given by the nature. Innate immune response is given by the nature, so that means you are not able to understand whether this pathogen is harmful or is not harmful to our body. So this will be decided by our body cells. Some of the cells that are already given, or the some of the environment or the medium that are already provided by the nature to protect ourselves, if you are not able to aware whether this person is harmful for ourselves and not harmful for ourselves. So this type of the response is the innate response. However, if we are knowing this response already, and this is the acquired response after the exposure, when you are going to know that this is the devil, this is harmful, this is pathogen, and this is harmful for our body, then after that, after that process, that after knowing by you, and the response that is achieved, this is called the adaptive or acquired response. So innate response defense which we are born with, it is the birth born, and it is birth born as well as. So what are the types of the innate response? The phagocytic cells, mainly the phagocytic cells that are WBCs. Two types, the microphagocytic and macrophagocytic. Microphagocytic is the most abundant, that is neutrophils, active neutrophils, that is microphagocytic in nature, while the macrophagocytic is the monocytes that are formed in the tissue. When monocytes pass in the tissue, then they are known as the macrophage. They are the macrophagocytic cells. And the another mechanism of the innate response is the complement proteins. Complement proteins, what they do, they coat the cell. They coat the cell that this cell is non self cell, and this is pathogenic for our body. So the complement proteins are released and complement system is achieved, and this complement system have the different criteria, different ways to kill any pathogen or any toxin, to eliminate this toxin, to detoxify this toxin. And as well as there is the anatomical and physiological barriers are already involved in our body. So these all type of response is the innate response. Like anatomical barrier is mainly our skin, that is not permeable for the large pathogen, and skin is also helpful in the thermoregulation. Physiological barrier, anatomical barrier as well as physiological barrier. However, main physiological barrier is mucous membrane, saliva present in the mouth cavity, oral cavity. Mucous membrane also present in the respiratory tract. So we are protected from by the help of mucous membrane, and mucus and secretory secretes from our body to protect ourselves from the digestive tract infection and the respiratory tract infection. So these are the physiological responses. Coming on the adaptive and acquired responses, the defenses that develop with the exposure and time. As the time proceeds, as actually the pathogen arrived in our body, so this response is delayed response. This response is not so fast. This response is slow response, and but this is more perfect response and more appropriate response. Adaptive acquired response is the slow response, but it is more appropriate and more perfect, and it is more able to diagnose whether this cell is our cells or the different cells. Sometimes innate response is not, however, innate response is very fast in nature, as well as it is not differentiating our cells and the foreign cells in very large prospectus. So, what are the responses? What are the adaptive and acquired responses? That are the serum antibodies and the T cells, that is cytotoxic T cells or cell mediated immunity. These T cells are mainly responsible for the cell mediated immunity, while innate response are mainly responsible for the humoral immunity. But adaptive acquired response are also can be humoral immunity. We are going to discuss further. So this is the first line of defense. First line of defense is the innate immunity, and second line of defense or third line of defense, that is adaptive and acquired immunity, because it is the late response, right? Mm, it is late response, but it is for prolonged period of time. Mechanism of the immunity. What is the mechanism of immunity? That is cellular level and the humoral level. Mechanism of immunity may be responsible by the sum of the cells of our body that are giving the immunity response. However, some of the humoral response. Humoral response means cells are not directly involved in the humoral response. Some of the cells, like B cell lymphocytes, they get active and form by the help of the complement system and different interleukins, tumor necrosis factor, and they get activated the plasma cell is able to form the antibodies, and these antibodies are mainly responsible for the humoral response. So this is the mechanism of humoral response. However, cellular response, cells are directly killing or phagocytosing the foreign pathogen. So this is the cellular response and this is the humoral response. Cell responsible for the protection is cellular response, and the cells that are mainly involved is the T lymphocyte and the phagocytic cells, like the or the natural killer cell, phagocytic cells or the natural killer cells, that are known as, also known as the natural killer cells. The 10% response is given by the natural killer cells. 10% immunity is provided by the natural killer cells, and they are the first responding, first the neutrophils, then the natural killer cells. Humoral antibodies are responsible for the protection, you all know. So this is the way. How two arms of the immunity. What is the two arms of the immune system? First, first part is the innate immunity, and second part is the adaptive immunity. Innate immunity is mainly responsible by the process of the phagocytosis, phagocyting the particles, and the adaptive immunity mainly responsible by the lymphocytes. Adaptive immunity is the slow in nature as well as it is for prolonged period of time, but innate immunity is the phagocytosing process. So, what are the phagocytic cells? I already told you that the neutrophils are the microphagocytic cells and the macrophages are the macrophagocytic cells that are responsible for the innate immunity. However, the criteria of killing the pathogen is the different. Actually, the neutrophils get burst on self and release the oxygen radicals. Release the oxygen radicals that are mainly. What are these radicals? These radicals are mainly, if you want to know, these radicals are mainly, suppose O2 minus, O2 minus, that is superoxide, O2 minus 2, that is peroxide, and that is another, that is, these are the superoxide, peroxide, or the free radical, oxygen free radicals. These are mainly responsible for killing the pathogen like bacteria, viruses, fungus, small fungus, or the hyphae of the fungus. These all are the non-self pathogens that are entered in the body and harmful for the body. So these all are killed by the neutrophilic cells, and these neutrophils actually have the granules. There are two types of the granules in the neutrophils: generalized granules and the specialized granules. So, specialized granules are the enzymes mainly. These enzymes are myeloperoxidase and the different superoxide mutase. So, what are these? Are the radicals? These are the oxygen free radicals and or the oxide ions that can kill the pathogen or the lysis of the cell membrane of the pathogen, and hence get cell pathogen get burst. But everyone wants to live for quite a long period of time. So at that case, what happens? What happens? Pathogens also produce some enzymes that can that can dissolve or that can detoxify these oxygen free radicals or these harmful oxide ions. So what are these enzymes? That are superoxide mutase. Have you heard about the superoxide mutase? What it does? Superoxide mutase convert the superoxide ion into the less harmful oxide ion, that is the peroxide ion. In the same sequence, peroxide ion get converted into the oxide ion, and hydroxide ion get converted into the water. And water is not harmful for the pathogen as well as for our body. So these are the enzymes that nullify the neutrophils. Means neutrophils are not able to kill those pathogens that have the enzyme superoxide dismutase or peroxidase, hydroxylase. These are the enzymes that are produced by the pathogens to prevent himself from our immunity, that is mainly innate immunity. But the different mechanism of the macrophage, tissue macrophage, is more lethal because it engulf the whole pathogen instead of releasing the enzymes. So how it actually working? Actually, T helper lymphocyte are the three types: T helper 1, T helper 2, and T helper 17. Mainly three types. There are also different T helper lymphocyte. Mainly three types you have to remember: T helper 1, T helper 2, T helper 17. T helper 1 activate these chemokines and cytokines. Actually, these chemokines led to inform the neutrophils. Neutrophils are moving inside the blood vessels in the center of blood vessels. Remember, this question is already asked by the different exams, that where neutrophils are present in the bloodstream. Neutrophils are present in the bloodstream in the center of the blood vessels. How they are going to know where they have to come? So how they are going to come? Actually, at that in that process, the chemokines or the cytokines get released, that are interleukins 1, 3, 4. These interleukins led to make the membrane permeable and giving the signal to the neutrophils that there is the inflammation process and you have to come here. So how the neutrophils get moved? Neutrophils came outside the vessels, that is vasodilation. Vasodilation process occur, and there are the certain responses that vasodilation process occur led to by the help of the prostacyclin PGI2, that is prostacyclin, and certain nitrous oxide synthesis enzyme is active and forming the nitrous oxide, and that led to the basement membrane gets shrinkage, and there is the perforations or the more space is provided in the blood vessel. So that led to move these neutrophils outside the blood vessel, and by the process of the diapedesis, this cell came out in the tissue tissue environment or the external environment outside the blood vessel, and again by the process, by the help of the attractive chemoattractants are released, and these chemoattractants led to make the neutrophils reach to the process where the inflammation is going on, and this neutrophil get burst itself, releasing the oxygen free radicals, and that was the pathogen. So this is the process. But in that case, there are the lot of the prostaglandins released, because the cell membrane of every whether it is prokaryote or whether it is eukaryote, cell membrane is mainly made up of the lipids and the proteins, and the main lipid that is also palmitic acid, arachidonic acid, and arachidonic is the precursor of the prostaglandins, and prostaglandin is formed by the two pathway, that is COX-1 pathway and COX-2 pathway. COX-2 pathway is responsible for the leukotrienes in the lungs or in the somewhere else, but COX-1 pathway is mainly responsible for the synthesis of the prostaglandin PGG2, and from the PGG2 all the prostaglandins are formed: PGD2, PGE2, PG alpha, PGF2 alpha, PGF1 alpha. These are the different prostaglandins. So cyclooxygenase enzymes get activate in the response of the neutrophils when the macrophagocytic cells are activated, hence the inflammatory process arises, and that led to increase in the temperature and the pain as all these processes are going on. Then the adaptive, coming on the adaptive immunity, this is the lymphocyte. The two types of the lymphocyte, whether B lymphocyte and the T lymphocytes. You all know that B lymphocytes get mature in the bone marrow, T lymphocyte actually mature into the T lymphocyte, mature into the thymus. And what do you mean by the maturation? Maturation means what are the CD markers present in these T lymphocyte. whether it is CD3 positive or not. CD3, if the T lymphocyte is CD3 positive mainly, both T lymphocyte and B lymphocyte have the CD3 positive, but the main mechanism to differentiate T lymphocyte and B lymphocyte by the help of the different CD markers. T lymphocyte is can be double positive or the double negative or the only one positive. So there is the mechanism. CD4 and CD8 both are present in the T lymphocyte, then this is the called the double positive. And if only CD4 is present, that means it is the T helper cell, while if it is the CD8 positive, that means it is the T cytotoxic killer cell. And coming on the B lymphocyte is having the CD19, CD20, CD35. These are the CD markers present on the B lymphocyte. Actually, CD19 is continuing by the help of the lymphoblast. Mm, if you are going in the case of the medicine, when you are going to study the internal medicine, in this internal medicine topic, you are going to know about the lymphoblast. There are different mechanism of this formation of these lymphocytes and the neutrophils, basophils, and eosinophils. These all are formed by the myeloblast cells, lymphoblast forming the lymphocyte, erythroblast forming the erythrocyte, and this is the process going in the bone marrow. By these all pluripotent cells get converted into the further subsequent cells, that is a mature form. So CD19 is present in the lymphoblast. CD19 is especially present in the lymphoblast, and except CD19, there are also present CD11 and CD64 and CD33. Here CD11, CD64, CD33, and also we can differentiate the B lymphocyte. Uh, B lymphocyte. So CD19 is mostly present in the B lymphocyte, as well as CD20 are also appear when they get mature. So CD20 is the most characteristic form to differentiate the B lymphocyte and T lymphocyte. And T helper 1, T helper 2 cytokines led to the formation of the B lymphocyte was T helper 1 and T helper 2 cells giving the response to form the antibodies. Right. Coming on the further, coming on the further cell of the immune system, that is myeloid lineage cell of the innate immune system, that is tissue macrophage and circulating neutrophils. Circulating neutrophil is formed by the myeloid cell. There are the different type of the myeloid cells. Actually, myeloid lymphoblast are of different types. Myeloid lymphoblast may be M2, M3, M2 or M3 mainly. Myeloblast if cause the cancer in the like AML, acute myeloblastic leukemia. In this case, M2 and M3 is responsible. If M2 and M3 both can be responsible, and the characteristic shape and size is the large. These myeloblast cells are large, as well as they have the granules, and the nucleoid is nucleoid. Remember, not nuclei. Nucleoid is well prominent, and granules as well as the azurophilic granules are present in these myeloid lymphoblastic cells. So this is the characteristic diagnosis of these appearance, right? And how they are going to differentiate in the cytochemistry and the immunophenotyping. In the cytochemistry, you are going to diagnose it by the Congo red dye, and with that Congo red MPS positive means myeloperoxidase positive. It is the myeloperoxidase positive. Which one? Myeloblast cell. So myeloblast cell, if these are the characteristic features, then you can say that it is the M2 or M3 type of the AML, acute myeloblastic leukemia, while M4 is for the monoblast. Monoblast, if the monoblast amount is much more as comparison to the normal, then this is called the M4 type of the acute myeloblastic leukemia. Right now, I am not going in the internal medicine. I only want to tell about the immune system. So B cell forming the antibodies by the help of the T helper cells, and T helper cells giving the signal to the B cell as well as the cytotoxic T cell. Cytotoxic T cell kill the pathogen directly, while B cell not killing the pathogen directly. They are forming the antibodies, and antibody mediated the killing of pathogen. But also remember, antibodies are not directly involved in the killing. So this is the case of the lymphocytic stem cells. Coming on the macrophage, macrophage is the tissue macrophage that is formed by the monoblast cells. So this is the phagocytosis and activation of the bactericidal mechanism, antigen presentation. What are the functions of the macrophage? That is phagocytosis. It can cause phagocytosis first, and macrophagocytic, right? And also cause the bactericidal, means kill the bacteria, and after killing the bacteria, also it present the antigen on the cell membrane. Antigen on the cell membrane, and from that B cells came, plasma cells came, and when they detect this antigen, and in that response they are forming the antibodies. Remember, these all cells are measure histocompatibility 1 positive. uh, you have to remember that all the cells of our body are measure histocompatibility complex 1 positive, except which one? RBC. All the nucleoid cell, all the nucleus cells, all the cells which have the nucleus, they all are measure histocompatibility complex with platelets. Platelets are also positive for the measure histocompatibility 1, because platelets already have the nucleus, but this nucleus get fragmented out. However, in the RBC there is no nucleus. In the mature RBC there is no nucleus, so that's why RBC are measure histocompatibility 1 negative as well as measure histocompatibility 2 negative. But there are the certain cells which have both positive, measure histocompatibility 1 positive as well as measure histocompatibility 2 positive. How? Measure histocompatibility 1 is already present in our body, as I already said that, but measure histocompatibility 2 is present in these cells where the CD4 marker, CD8 marker is present. Remember, so what are the characteristic feature of these cells? This is the large cell and have the diameter about 10 to 25 micron meter. Purpose is main phagocytosis mediated killing, and also this is the non, remember it is the innate type, so it is the non-specific immunity. Non-specific, while specific immunity is acquired type. So it is non-specific and chemotactic capability, potent phagocytosis when activated by T lymphocyte. So there are the lymphokines that release and led to the phagocytosis. And after this phagocytosis, it also provides the antigen to form the antibodies for the future, and also it led to formation of the memory T cells. Memory T cells, suppressive T cells, regulator T cells. These are the response. And coming on the neutrophils, these are the neutrophils, phagocytosis and bactericidal mechanism. Both are mechanism. However, the characteristics of all these neutrophils, you all know that multilobulated nucleus is present and neutrophils are already formed from the myeloblast cell. Precursor is myeloblast cell. Multilobulated nucleus, lysosomal granules is present, and they have the specific granules as well as the non-specific granules. Phagocytosis and killing process, and this is the first white blood cell to infection site. This is the neutrophil. Remember, neutrophil is small, and that's why it have the different, also different characteristics. I'm not going in these details. I'm going to this detail in the internal medicine when we talk about the mainly cancers by caused by these cells. So white blood cell to infection cell site and die and release content. They are killing the pathogen. They have the bactericidal nature by the releasing content, and the releasing content is mainly oxygen radicals or the superoxide ion or peroxide ion. Remember, these releasing contents are not applicable and not able to kill the pathogen in case if the bacteria have the enzymes like superoxide dismutase or the peroxidase. Coagulase also. Irritant surrounding tissue recruit cells. Phagocytosis improved by the opsonization with IgG antibody. For this killing, it is able to respond for those bacteria or those pathogens mainly where the antibodies are already get opsonized. Opsonization means antibodies get engulfing any pathogen or antibodies surrounded this pathogen or coding this pathogen. Coming on the circulating neutrophils, So these are the factors. What are the factors? Interleukin 1, interleukin 6, as I already told you, interleukin 8, tumor necrosis factor. These all release, when these all release, that led to the extracellular bacteria get killed, and there is the no memory or the permanent cells developed. So inflammatory neutrophils consist of these granules, and this bacteria get engulfed together and get killed, and in after that killing there is the antigen that get present in the cell membrane of these cells, right? So this is the inflammatory response. Bacteria triggers macrophage to release the cytokines and chemokines. After releasing cytokines and chemokines, there is the vasodilation process occur, as I already explained you. Vasodilation process occur, and through the diapedesis process they move outside the blood vessel, outside the blood volume, and move in the tissue space and tissue fluid, and they go to the site where bacteria is present, and they engulf this. Inflammatory cells migrate in the tissue and releasing inflammatory mediators. Inflammatory mediators, I already told you, when this cell membrane get ruptured, this release arachidonic acid, cyclooxygenase enzyme activated, and the prostaglandins are formed. So basophils, coming on the basophils, these are the poor at phagocytosis. Granules, what are the granules that contains basophils? That is histamine, serotonin, and vasodilators permeability factors. Basophils are mainly responsible to make the blood vessels permeable and kill this response and kill the bacteria, and also require binding of the two IgE antibody release. If you want to know about the Ig antibodies structure, mainly this release the histamine, serotonin, and the different form. So mainly, if you want to know, that is the sorry for the inconvenience. How it is working? Actually, this is the antibodies. This is the antibody, and the response of this is here. Each antibody have the two chains, light chain and the heavy chain. So two heavy chain and two light chain. There are four tetrapeptide chains, and the molecular size, molecular weight of these chains are mainly responsible to determine the total antibody molecular weight. And remember, these all antibodies are classified on the basis of their heavy chains. On the basis of their heavy chain. For example, alpha chain is present in the IgA antibody. Mu chain is present in the IgM antibodies. Gamma chain is present in the IgG antibodies. So that are the different form. Delta chain present in the IgD antibodies. Epsilon present in the IgE antibody. So in the same sequence, heavy chain is responsible to differentiate this 5 class antibodies, and while the light chain, between the light chain and heavy chain, there is the two region, that is the constant region. Here is the constant region, constant, constant region, while there is a variable region. Variable region have the paratoph, and it bind to the epitope of the antigen. Antigen have the epitope, and paratoph region is present in the antibody, and there are the two variable region in the both side, right? So there are the two paratoph, variable region or paratoph region, they same meaning. So when paratoph join to the epitope, hence antigen get attached to the antibody, and every antibody have the different paratophs. And also there are the sulfide linkage present between the two heavy, between the heavy chain and the light chain, and there are the different enzymes that came and lysis of these antibodies appear. For example, pafsane, trypsin, and the different proteolytic enzymes can get break down the heavy chain and the light chain. And so this is the called FC portion and FEV portion. FEV portion is the large portion and FC portion is the small portion. So if we talk about the, for example, pafsane break down the any antibody into the two FAB part and one FC part in the same way, right? So coming on the phagocytosis, so this is the mast cell. Mast cell, what is it does? Mast cell release the histamines and other active agents. So basophils present, uh, actually in the blood. These cells are known as the basophils, and when these basophils get move outside the blood vessels, then these cells in the tissue is known as the mast cell. After that, mast cell, you can differentiate that resting mast cells are the granules. These granules are getting released. Here it is the activated mast cell, and here the antibodies get suppressed, secreted out in the Ig antibody form. Coming on the lymphocyte is the very wide range. Lymphocyte is the small lymphocyte up to the large lymphocyte and the medium sized lymphocyte. Small lymphocyte have the another characteristic feature and other significant importance if you want to diagnose the microcytic anemia. Microcytic anemia means you are talking about the anisocytosis or the MCV phase of the characteristic mean corpuscle volume and the mean corpuscle hemoglobin concentration. If you want to diagnose this or the anemia in the patient, or you want to diagnose the size of the RBC, then you have to compare the small lymphocyte to the size of the RBC. If the size of the RBC is less than this small lymphocyte, then this is the microcytic anemia, MICRO, microcytic anemia. So, but the lymphocyte have the no lysosomes, and it is known as the brain of this immune system. Until activated, it mainly regulate all the cells, all the innate cells as well as the innate immunity as well as humoral immunity. So this is called the brain. Distinguish the self and non-self. Distinguish it from the self and non-self, and recognize specific antigens, and also the memory cells are also forming from these lymphocytes. And need presentation of the antigen by the macrophage. Antigen must be present by the macrophage, because when I remember when macrophage get phagocytosed and pathogen, then this also present the antigen. So antigen must be present to form the antibody.